All right, so good afternoon. This is a video that's going to review your unit over genetics to prepare for your test coming up. And so just real quickly, I wanted to show you when you look at a study guide question and to be the best and have the best quality information on a study guide, I kind of want to show you question number 1 says describes Mendel's studies and conclusions about inheritance, and to show you the difference between an okay answer and then a really good thorough answer. So when you look at Gregor's Mendel studies, you could say he studied pea plants, and then he studied 3 tests. He did 3 different tests, and he did a troop breeding and cross-pollination, and he made up 3 laws. That's okay. That's answering the question, but you're not really getting a full quality of information. So over on this side would be what you maybe would want to include instead. Gregor Mendel studied pea plants. He used peas because they were easy to grow. So why did he use pea plants? What did he study with those pea plants? He used and looked at 7 traits. He completed 3 different tests and naming those tests. And then what did he discover? What was his conclusions about inheritance? Well, he discovered 3 laws and then named those 3 laws. That would be the best, most detailed to just answer question number 1. So to be successful in the test, using your packet and really thoroughly giving a good, substantial amount of quality answers to fully complete the question would be the best when studying and preparing for your test. So anyways, looking at Gregor Mendel, who was he? He was a monk in Austria who studied peas. And he studied pea plants because they were easy to grow. You could get quick results. You were able to see a lot of offspring very quickly. And so he studied these pea plants and noticed there was a pattern. There was a pattern of something from the parents in these pea plants. When they had baby pea plants, they were passing something down. And so he contributed to this whole idea of inheritance and passing down something from parent to offspring. And genes and genetics wasn't really the terms they used back then, but he was the father of genetics leading that pathway. So just some vocabulary to define. So we have an allele. So an allele would be what I'm circling. And so that letter C would be an example of one of the alleles in your genotype. And that's a great another thing is what is a genotype? And so that would be if you have your full complete letters together. And then phenotype would be what you physically see. So green pod color would be an example of what you physically see. And then we have heterozygous genotype. That would be when you have two letters, one uppercase, one lowercase, to describe your genotype. Homozygous, you could either have homozygous dominant or homozygous recessive. For example, little y is homozygous recessive. Big R would have been homozygous dominant. Then sex-linked is a trait. That means it's on the sex chromosomes, the trait and the gene is. And so this is how you would write genotype for sex-linked. You'd have either an X and X for female or an XY and then a letter at the top to show that. And blood type is written in this format. You can have A, B, or O blood, and you have 2 alleles for your blood, but there's 3 choices. And so this would be type A blood, and you write it that way. He has an A allele and an O allele. So it's capital IA and then lowercase I for O. That's just referring to back in our unit. So to just briefly discuss about chromosomes, remember that you have chromosomes, and on these chromosomes, these bands are these genes, and each gene has an allele. So when you look at, when we say homologous chromosomes, we're saying that in our body, we have 46 chromosomes, and they're homologous, which means we have 23 pairs, which means we have 23 chromosomes that are from our dad, and we have homologous match from our mom. So we have 23 pairs equal a total of 46. Now, when we go through S phase and we make a copy to get ready to make our gametes, our sex cells, they are duplicated. So then you see that dad's was replicated and has an exact copy attached with the centromere, and then the mom's has an exact copy attached with the centromere. All right, so going and talking about Mendel's laws again, looking at the patterns of inheritance he saw, this picture is showing you the law of independent assortment. So what we're saying is that when these alleles segregate, so when this G segregates and goes into this sperm, and this G segregates and go into this sperm, they're going to separate independently of each other. So notice that the position of where they are located are like this, and they might be different in a different situation depending on when the gamete is formed and what happens. So maybe this chromosome actually was down here instead, and so then it would have gone in a different sperm choice later. And so that's what the independent assortment means, is they separate independently of each other, and you actually don't know really which alleles will show up in your sperm. So this is the law of dominance, and you can see that the dominant trait is represented with a capital letter. So for example, capital R is referring to this purple, and you can see that lowercase r's mean white. And so if you get a big R, little r, you're showing that this is dominant over the white, and so you see purple. That's the law of dominance, and not everything is following the law of dominance rule, and we'll get to that in a moment. This is just showing you the simple law of segregation. Now this is not as specific as the law of independent assortment. It takes it a step further in that law, but this law is just stating that you have two alleles, one from for each gene. So you get one allele from your mom and you get one allele from your dad, and that makes up one gene. Now when you make your sex cells, the alleles will segregate. So one allele will go in here and one allele will go in here. Now the law of independent assortment takes it a step further, but this is more simplistic. All right, so we looked at Punnett squares and probability, and so this is a dihybrid. This is a di, meaning two traits are being looked at the same time. So for example, in this dihybrid, we're looking at shape and we're looking at color of pea plants. And so how many gametes form from a dihybrid? And you should say it's due to independent assortment. And remember, so this would be the mom, this would be the dad, and this would be their sperm choices, their gamete choices. So you get 4. And so if you looked at this, looking at how many different phenotypes are in this Punnett square, well, we see round and yellow, round and yellow, round and yellow, round and yellow. And if you look, all of them are one phenotype. So there's actually only one phenotype in this Punnett square. This was an easy one to analyze, and it only has one genotype difference as well. Both of the traits are heterozygous. Capital R, lowercase r, capital Y, lowercase y. So when we talk about exceptions to the rule, there are other patterns of inheritance that don't follow the law of dominance. In this one, you see a red flower and a white flower crossing that have homozygous dominant genotypes. When they get together, they have a heterozygous genotype, but instead of seeing red or white, we see a pink, we see a blend, and that is called incomplete dominance. You remember the word blend when you think of incomplete dominance. This is an example of when you see a white chicken with homozygous dominant, and then you see a homozygous dominant black chicken, but when you come together, they're heterozygous, but you should see black and white. That is what we call co-dominance. They are co-dominant together, so you should see both phenotypes equally, and we think of checkered or spotted or speckled or something like that, so you should see both phenotypes. Now this is looking at blood type. A blood has either 2 A's or an A and an O. B blood can either be 2 B alleles or a B and an O allele. So you notice that A is dominant over O and B is dominant over O. But then when you get to AB, there's an A allele and a B allele, and you become an AB blood type. And then if you're an O, you should have 2 little i's. So this is how we represent genotype for blood type, and go back to your Punnett square practice problems to notice that, but this is a multiple allele pattern of inheritance as well, where you have an A allele, a B allele, and an O allele. And so you might want to go back and refer to your Punnett square practice problems to figure out what the probability would be of offspring having A blood with these parents. You should see that there would be 25% chance. All right, so this other pattern is called sex-linked. And we notice this is a sex-linked because I talked about color blindness being a sex-linked trait. You can see that we're looking at X's and XY's. And you can see that the mother has 2X. So this is what we call a carrier, where she has the dominant allele, but then she has the recessive color blindness allele. So she's a carrier for it. So she has the potential to pass it down. So please refer to your Punnett squares reviewing sex-linked practice problems as well. So eye color. eye color, look at all this variation in phenotype. When we look at eye color and we look at polygenic traits, we should see a bell-shaped curve where kind of on average there is more in that number, but then there are some, like for height, an average height would be up here, but really shorter people and really tall people would be on the outsides of your bell curve. And this is called a polygenic trait. Many, many genes contribute to this trait, and that's why you see such a variety of variations of phenotypes. All right, so moving into meiosis, there is something that is really important that happens in meiosis when we make our gametes, and that's during prophase 1. In prophase 1, a process called crossing over happens, where these homologous pairs form a tetrad, and they sit right on top of each other, and they actually switch genetic information. It only happens in prophase 1. When you look in prophase 2, it is not happening. So crossing over only happens in prophase 1. And then we go through 2 processes. We go through PMAT 1, and then we go through PMAT 2. And so for example, if you look at this situation of meiosis, this would be anaphase 1 because the entire whole chromosome is moving away. But down here, the chromatids are moving, actually separating from each other, and they're 2 cells. That would be a difference between anaphase 1 and anaphase 2. So comparing meiosis to mitosis, both of them start with 46 in their cell. Both of them have replication that occurs, so now they have 92. But the difference is that in mitosis, you go through PMAT once, and you get 2 diploid identical cells that you started with. But in meiosis, you go through PMAT twice, and so you end up with 4 haploid cells, 23 in each cell instead. And so there can be problems that arise in this when you're forming your gametes, and that's why we looked at karyotypes, because something called non-disjunction can occur, where the chromosomes don't separate correctly, and you either might get too many chromosomes or not enough chromosomes in your gametes. So this is a karyotype, and we practice looking at these. And so the sex of this karyotype would be a male. It has an X and a Y, but it actually has 2 X's. So it actually is an abnormal karyotype where you have 47 chromosomes. If you counted each individual chromosome, you would get 47, and that's abnormal. And so you have a 3rd chromosome right here on the X chromosome that you're not supposed to have. So we call that trisomy, when you have a 3rd extra. You're only supposed to have 2 sex chromosomes, and you have 3 sex chromosomes. So that would be something called trisomy. All right, and then lastly is pedigree. So you need to analyze a pedigree and be able to understand what it's reading. So this is looking at 3 generations. So this would be generation 1, generation 2, and generation 3. Now, squares are males and circles are females. And if it's shaded completely dark, that means they have the trait the pedigree is looking at. And so if you look at 1 and 2, this line here is showing that they have had children. And so the relationship between 1 and 2 and down here is that these are the children of person 1 and person 2 right here. If you look at this circle, number 3, 4, and 5 right here, these are siblings with each other that 3 and 4 had. And so when you look at the genotype of trying to figure out the genotype of something, you automatically know, let's say we'll just use lowercase p. This is automatically going to be a lowercase p because it doesn't have the trait. It's not shaded. And this is something that you would notice that if I would be very specific on whether I said, and it says it's an autosomal dominant, so you have to have a big letter to have it. So we know that 5 person automatically got one of these. So what do you think person 1 is? Well, we know that 5 has to have a dominant allele, so because he's shaded. So we know that dad has a big P, but does he have another big P or does he have a little p? The answer is he has a little p, and we know that because we have to look at his kids. This kid doesn't have the trait, and number 6 doesn't have the trait, and so he had to have given this little recessive allele to his son here, and then the mom would have given her little p here, and that's how you can determine what the genotypes are. You really have to look at their kids and what they have. All right, here's your root words. Don't forget about them. And remembering root words and trying to prepare for your root words. I might bring some old ones back that we've used in the past: hetero, homo, meta, pro, those kinds of And so to be successful, read over chapter 11 and 14, go over your Punnett squares and your practice problems, and don't forget about your study guide.